Hi, in this video we'll talk about the M phase of the cell cycle, which focuses on mitosis, which is the separation of chromosomes, and cytokinesis, the division of the cellular material or cytoplasm. We'll end by talking about some of the forces involved with chromosome movement, both the inward movement to get the chromosomes to align in the center, the metaphase plate during metaphase, and then also the forces responsible for separation of the sister chromatids with one set of chromosomes going to each daughter cell. Just to emphasize, we're talking about the M phase, in which the duplicated DNA material is separated and divided up between daughter cells. So here we see the overview, IPMAT, interphase, prophase, prometaphase, metaphase, anaphase, and telophase. And we have one slide for each of these phases and a discussion of some of the mechanisms that occur during those phases. This is approximately a 1-minute animation with no volume, but it's a good visual depiction of what's going to go on during mitosis. And then in prophase, two important things are happening. One, you have the condensation of the chromosomes, and secondly, you have the duplication of the centrosomes, which together the centrosomes, the microtubules, will form a structure called the mitotic spindle, also called the spindle apparatus, which is going to be responsible for forced generation and chromosome and cellular separation. We've talked a little bit about chromosome condensation earlier, primarily at lower levels with DNA being wrapped around histones. Now we're talking about higher level compaction, which involves proteins called condensins to help pack loops into ordered structures, and the related proteins cohesins, which will help keep the two sister chromatids connected together during various stages of the process. We also see here the mitotic spindle formation, a centrosome being duplicated, the microtubules pushing against each other, driving the two centrosomes apart. Now you have a large structure called the mitotic spindle, and the microtubules in the center here start to act with the chromosomes, binding to the kinetochores proteins at the centromere, pushing them initially to metaphase to line up in the midpoint between the two poles of the mitotic spindle. The centrosomes are now called spindle poles. In prometaphase, the key event is the dissolution of the nuclear membrane. As we'll see, this is mediated by phosphorylation of the lamin proteins and the nuclear pore proteins. With the nuclear membrane gone, now the microtubules are free to interact with the chromosomes, primarily interacting at the centromere, which will initiate the aligning of the chromosomes at the midpoint between the two spindle poles. Basically, giving a little visual detail on what we just talked about, let's just talk about the chromosome here. These are sister chromatids, copy 1 and copy 2. Let's say this is your maternal copy of chromosome 1. Now you have 2 copies of the maternal chromosome. One will go to each daughter cell. Elsewhere, you have the chromosome 1 that you inherited from your father, and those will also be duplicated, so you'll have 2 copies of your paternal chromosome. So right now you have 4 copies of your chromosome 1, 2 from your mother, 2 from your father. At metaphase, you have the chromosomes aligned in the center between the 2 spindle poles, and now you have attachment of each chromosome to each of the spindle poles. During prometaphase, when the chromosomes were still moving to the midpoint, these chromosomes were attached to the spindle pole closest to it, which is why it's getting pushed to this midpoint. Now when you have microtubules connecting from both sides, you have a balancing of forces, and that helps align the chromosomes at the midpoint. To get from metaphase to anaphase, where we're starting to separate the chromosome, a couple things have to happen. One thing that happens is this protein complex called APC, which is the anaphase promoting complex, gets activated, and this has proteolytic activity. Some of the proteins that it's going to degrade is the cohesins, which keep the two sister chromatids connected. It's also going to destroy M-cyclin. Remember, that is critical for the exit of the M phase of mitosis and promoting the metaphase to anaphase transition. But before this degradation of the cohesin proteins can occur, we're going to look at the checkpoint and how it makes sure that each spindle pole is connected to one of the two sister chromatids. If everything checks out at anaphase, the sister chromatids will start to move to their respective spindle poles. This occurs by a shortening of the microtubules that connect to the kinetochore, because the microtubules are being degraded here at the plus ends of the filaments, with tubulin subunits being released from the microtubule. A second thing that's happening is these two spindle poles are starting to move further apart. They may start out at, let's say, this sort of a distance, and then by the end of anaphase they have reached a larger separation distance. And this occurs here, if you can look in this one, by the two microtubules pushing against each other, which has the net effect of forcing the spindle poles further apart. And then in telophase, they are now separated by a large amount. The nuclear pore proteins in the lamins underneath start to reform the nucleus as a organelle, and a contractile ring of actin and myosin form at the midpoint between the two spindle poles and will start to pinch on the cytoplasm, promoting cytokinesis. On this slide, we see the manner in which the nucleus is first degraded by phosphorylation of nuclear pore proteins and the lamin proteins, the intermediate filaments which give the nucleus structural integrity. These proteins are not degraded at all but maintained, and then when a phosphatase will cleave the phosphoryl groups off the proteins, this will cause the proteins of the nuclear pore and the lamins to reaggregate and start forming a new nuclear membrane during telophase. At the end of cytokinesis, the contractile ring made up of actin and myosin is about to pinch off the cytoplasm. This will sever the microtubule connections from the spindle pole. The centrosome will start to reform the normal structure that exists during interphase. The chromosomes will start to decondense with the cleavage of the condensin protein and will start to get dispersed interphase chromosome structure, which is now entirely contained in a reformed nuclear membrane. There's not too much new here, except to say that the contractile ring, which is dividing the cells, is perpendicular to the long axis of the spindle pole, and it's also here at the midpoint, forms at the midpoint between the two spindle poles, which is very useful because while it's regulated that each daughter cell gets the equal set of chromosomes, things like mitochondria, which are stochastically separated, which is to say that there is a lot of them present before cytokinesis, and it just so happens that if you cleave right in the center, both cells will get roughly an equal amount of those, but there can be slight variations. And this is a slightly interesting electron micrograph where you see the structure called the cleavage furrow, and this is where actin and myosin are contracting in here on the plasma membrane. So on to the description of the mechanisms of chromosome movement. Again, we have these duplicated centrosomes forming what the overall structure called the mitotic spindle. It shoots out a variety of different microtubules. There are the blue kinetochore microtubules, which will attach to the kinetochores. For chromosomes that are initially closer to this centrosome, they'll be just simply by chance, they will get connected to a microtubule from the closer spindle pole. Eventually, it will just be pushing out the chromosome until it has a chance of hitting another one, another microtubule originating from the other centrosome, and then a balancing of forces will naturally lead to a central location here for the particular chromosome. There are also the interpolar, between the poles, microtubules which will interact with each other, and there's motor proteins here which are going to try to slide the microtubules past each other. This will push and produce an outward force which is going to lead the two centrosomes to get further and further apart as division proceeds. Finally, there are the microtubules referred to as the astral microtubules, which help position the centrosome in relation to the cell membrane. These are obviously kept shorter for that, and then they will actually get shorter and shorter as these two centrosomes are pushed farther and farther apart. This slide here is showing an earlier stage where the sister-chromatids are still being moved out to the metaphase plate, which is the midline. And again, the fact that this chromosome was closer here, it is subtending a larger arc, so it's more likely to get hit by a microtubule that gets shot out in roughly random directions. Eventually it's going to make contact. The motor proteins here will move the chromatids towards the center, and eventually it will encounter a microtubule originating from the other centrosome. We also see these specialized kinesins, which are double-headed, which will walk along this microtubule in this direction, along this microtubule in this direction, which will produce a sliding force that producing this end of the microtubule going that way, this end of the microtubule going this way, again forcing the two centrosomes further and further apart. You also have here a dynein which walks retrograde on the microtubule pathways, and that's going to contribute to pulling the centrosome out as it's attached here to the membrane, but it's going to pull the microtubule in this direction by walking inward, with this end being degraded as needed. And during anaphase, with the separation of the chromosomes, you actually have two things occurring. One is the degradation of the microtubules by the kinetochore proteins, shortening the microtubules, so you have separation of the sister chromatids, and also you have the double-headed kinesins, which are pushing the centrosome poles further and further apart. They start out this far apart, and now they're this far apart. So I think it's rather interesting to see how very simple and mechanical the chromosome motion is during mitosis. And perhaps you lose some of that wonder, because when I was first studying biology, it just appeared that the chromosomes were flying around and somehow knew where to get. There wasn't even that much questioning about what was happening, but it can be very simply explained once a little of the molecular details are known. And here we see the mechanism, which is also highly mechanical, of the metaphase to anaphase checkpoint, which is basically going to measure that there is roughly equal tension in both directions for each chromosome. This is a tension test to make sure that each sister chromatid is attached to a different centrosome. This is going to help ensure that each daughter cell receives the full set of chromosomes. If only one chromosome is attached to a centrosome, if by chance both of the sister chromatids are attached to the same chromosome, you're going to have unstable situations where separation of the chromosomes will not be allowed to proceed. I'm not certain about how the tension is monitored by the cell, but there are plenty of proteins that change shape upon being physically stretched or forced, so that it's not hard to imagine an enzyme that becomes active or inactive based on a certain amount of lateral stress, and this could promote passage through the checkpoint and allow chromosome separation. And the last slide on this video talks about organelles which are not specifically separated, like the chromosomes, mitochondria, endoplasmic reticulum, and the Golgi fragments. And you can think of all cytosolic proteins and these other organelles as being separated relatively in half by chance, because the volume of the two cells is somewhat controlled for. Okay, thanks for listening.